Hello guys, welcome to the knowledge domain, the concept of life sciences in biotechnology. In today's lecture, we are going to discuss the electron transport system of the mitochondria, and there will be a comparative study on the electron transport system present in the chloroplast also, and how ATPs are synthesized. So mitochondria is an organelle which is a subcellular structure and having a specific function. As we discussed, one of the most important function is the generation of the ATP. That's why it is called the powerhouse of the cell, and it is also involved in the apoptosis. And because of the uncoupling, it is the source of the free radicals, the reactive oxygen species also. So this is the mitochondria having two phospholipid bilayers, and there is a space between them. This is called the intermembrane space. And the inner phospholipid bilayer that is invaginated into the matrix of the mitochondria, this is called the cristae. Okay. So this is a schematic for a better representation of the mitochondria. This is the outer mitochondrial membrane. Outside this is the cytoplasm, and this is the inner mitochondrial membrane that forms the cristae. This is the cristae. This is the cristae. And the electron transport system molecules are present in the inner mitochondrial membrane, and oxygen that is going to be used to form the water molecule is imported from the cytoplasm. The CO2 generated in the citric acid cycle before the ATPase that is moved out of the matrix into the cytoplasm, and the glucose oxidized molecule pyruvates as well as the fatty acids, they move into the matrix, and here the various molecules which are reduced like the NADH are present that will be used by the electron transport system to generate a proton motive force or a proton potential across its inner mitochondrial membrane. There is a high proton concentration here that will be discussed. Okay. So before discussing the electron transport system, we are going to see the various molecules that is going to be used in this, like the NADPase, that is the source of the electron, the energy-rich electron, NADH, the redox potential is 0.3 minus 0.320 volt, and the end product is the water molecule from the oxygen atom. So this is a not a balanced equation, but for one water molecule you need half of the oxygen molecule, and the reduction potential is 0.8166. So the reduction potential is increasing from here to the here. So the more reduction potential means the molecule present on the lower side of this table, they have the more ability to accept an electron. Okay. And if the reduction potential is increasing, means oxidation potential is decreasing. So the molecules present here, they have the tendency of releasing the electron. So that this will be discussed in one of the following slides. So remember that the NADH reduction potential is minus 0.320, and the reduction potential of the oxygen atom is 0.8166. And the various protein components of the mitochondrial electron transport chain is the component one is the NADPase dehydrogenase, where there is the iron sulfur molecule. The prostatic group is FMN, and the component second is the succinate dehydrogenase, F8 is iron sulfur. Ubiquinone, cytochrome C, oxidoreductase, cytochrome C are the component 3. The prostatic group is the heme and the iron sulfur, and component 4 is the cytochrome oxidase, and the group are the heme and the copper A and B. So this is the outer mitochondrial membrane. We can see this is the phospholipid bilayer. This is the inner mitochondrial membrane. We can see this is also a phospholipid bilayer. The space between the two mitochondrial membrane is known as the intermembrane space, and the lumen of the mitochondria, or the space inside the inner mitochondrial membrane, is called the matrix. So NADH is the source of the electron. So NADH releases the electron into the complex one, which is the NADH dehydrogenase, and oxidize itself to the NAD plus. Okay. So this electron, the two electron, it travels through the abandoned phosphatic group and a series of iron sulfur molecule through a process of oxidation reduction, and finally it reduces the quinone into the quinol. Okay, and it utilizes the two proton molecules from the matrix of the mitochondria. So during the process, the energy is stored in the NADH. This is released in a sequential way, and the energy utilized in pumping four proton molecules from the matrix of the mitochondria into the intermembrane space. Then the quinol here, it will releases itself from the complex one into the phospholipid layer of the inner mitochondrial membrane. Then the component 2 of the ITS is the succinate dehydrogenase. So succinate will be oxidized into the fumarate. During this process, it reduces FAD to FADH2. Okay, and the other molecules, they bypass the succinate dehydrogenase, like the fatty acyl coenzyme A. So fatty acyl coenzyme releases electron, and it goes through acyl coenzyme dehydrogenase, then electron transferring flavoprotein with the FAD, and finally the electron transferring flavoprotein with ubiquinone oxidoreductase. Finally, it is oxidized and it reduces the quinone into the quinol. Similarly, the glycerol 3-phosphate of the cytoplasm that is used by the glycerol 3-phosphate dehydrogenase in reducing the quinone into the quinol. So as I discussed in the complex one, the quinol diffuses itself into the phospholipid bilayer or the inner mitochondrial membrane. So this is the complex 3, and this is the cytochrome C oxidoreductase. So in this process, the reduced quinol, we can see here, that oxidizes itself and transfers its electron and reduces the cytochrome C into the phospholipid bilayer, so that after that the cytochrome C will move and bind into the complex 4. But during this process, the energy released, it is utilized in pumping another 4 proton molecules from the matrix into the intermembrane space. And the cytochrome C, it oxidizes itself and it reduces in half a molecule of the oxygen into the water molecule. So in this process, the energy released during this transfer of the electron from the cytochrome C into the oxygen molecule to form the water is utilized in pumping 2 proton molecules from the matrix of the mitochondria into the intermembrane space. So you can notice here the source of the electron in this process is the NADH, and the end product is the water molecule in case of the mitochondrial ATS. So if we make the equation balance, this is the complex 1 equation, NADH, the source of the electron, 5 proton molecule, N means the lumen or the matrix of the mitochondria, plus quinone. So NAD plus, this is oxidized from the NADH. Then this Q, this is reduced in the quinol, and the 4 proton molecules that are pumped into the intermembrane space from the matrix. This quinol, they are reducing the 2 cytochrome C1 oxidized. They are taking the 2 proton molecules from the matrix, and this is the finally oxidized quinone is here. And the cytochrome oxidase that is here, that is reduced in the cytochrome C, and the energy is trapped here And in this process, four proton molecules are formed from the matrix into the intermembrane space. And this cytochrome, two cytochrome C1 molecule is used in making one water molecules. Okay, so to finally make the equation complete, because water molecule. So the electron source is the NADH or FADH, and the end product is the water molecule and the proton gradient across the inner mitochondrial membrane. So this is we need to remember, because there is a difference on the electron donor and the final product of the electron transport system between the mitochondria and the chloroplast. So this is the total equation of the entire ETS, NADH, one molecule plus H plus oxygen atom equal to NAD plus, that is like before, and this is the water molecule. So during this process, the energy that is present as a chemical energy in the form of the NADH, so the electron that is moving from the low reduction potential to the high reduction potential of the oxygen atom. So whatever energy is present on this electron, it is expressed as the NF delta E. So this NF is the Faraday constant. This is the difference in the potential energy, means the reduction potential of both. So it is, that is the transfer of the two electrons. So this is 2 into 96.5 kilojoule per volt per molecule, and the difference, as I discussed in one of the previous slides, the reduction potential of NADH is minus 0.320 minus. So this is minus from the oxygen reduction potential, oxygen atom 0.8166 minus 0.320 equal to 1.14 volt. Okay, so total is 220 kilojoule per mole of the NADH. So this is the energy that is released during this process. So during this process, the proton pump is pumping, means during this process, the proton is pumped from the matrix into the intermembrane space. So the H plus that is making itself concentration in the intermembrane space results in the two types of potential. One, because it is a chemical molecule, so there is a chemical potential delta pH. So it is more proton, so positive charge, so this is acidic, so matrix is alkaline. And another is this is having a charge, so there is the electrical potential, so delta psi across the inner mitochondrial membrane. So next is, to further summarize, this is means it is in millivolt. Before we have discussed in the volt. So this is 3 minus 320 millivolt with the NADH, NAD plus reduction potential, means NADH reduction potential. This is oxygen atom reduction potential is plus point, means 816.6 millivolt. So this is series of, means oxidation reduction process, in which the energy released in the series of oxidation process is used in pumping proton from the matrix into the intermembrane space. So this is a plant system. In there is another source of the NADH, that is the 2-glycine. So they are reducing the NAD plus to form the serine and to carbon dioxide molecules, ammonia and the NADH. So this is an alternate source of the NADH in the plant. So the proton pump, the proton gradient that is generated, it maintains a electromotive force or the proton motive force, and how the ATP are generated on this process. So this is the P means intermembrane space, and the matrix. So because of this proton pump, there is a proton motive force that is generated here, and delta G, the free energy, equal to R is the gas constant, then T is the absolute temperature, and F is the Faraday constant. This is the F. Z is the number, the charge of the proton molecule, that is 1 here. So it is 2.3 RT delta pH plus delta pH means the change in the pH across the inner mitochondrial membrane, and delta psi is the potential, electrical potential across the inner membrane. So now it is generated and the membrane is impermeable, but this proton can move through this ATPase, which is also called the F0F1 complex. It has this has a pore here, you can see. This is the C. It is a complex protein complex of the proteins type of the C. And this is the actin filament that is attached to the cytoskeleton. This is the avidin. So this is holding as an anchor. This A protein, B protein, these actually make it immobilized. So this one and this axis, this will move. So there are the three molecules of the alpha and three molecules of the beta. And this alpha and the beta molecules in the F1 complex means they form one unit, and there are three forms of it. So the three forms are known as the one is the ATP, means it is a tight ATP binding form, means conformational form. Another is the loose ADP plus inorganic conformation, and the third one is the beta empty ATP conformation. So when a proton molecule passes, means three proton molecule passes, then the ATP conform, at least each of them will have this conformation in one rotation of 120 degree. So when the ATP is released from here, as long as this ADP plus inorganic phosphate does not bind tightly to form the ATP, and at the same time there is a ADP plus an inorganic phosphate is recruited and loosely associated, there will not be any movement of this means complex, this protein complex of the six along means on the axis of the gamma protein. So once this is empty, when it will be empty, at the same time the ATP plus ADP will tightly bind to form the ATP, and there will be a recruitment of a ADP molecule, inorganic phosphate molecule for the loose association. So these three conformations are stabilized, and there will be another 120-degree rotation because of the passage of the three protons. So again, the tight ATP will be released. The ADP plus inorganic phosphate, they will be combined to form the ATP, and a new ADP and an inorganic phosphate will be recruited for the loose association. So in each rotation, this event is repeated. So in each rotation, there will be one ATP molecule released into the matrix because the proton is moving from the intermembrane space into the matrix. So this is the proton stoichiometry. So this is the ATP, this is the inorganic phosphate, because each of them will bind to from the X number of the ATP molecule. And this is an atom of the oxygen. This is the H plus, this is the NADH. Okay, so this is the X number of ATP from here plus one molecule of the water, because this is only one atom, and this is the NAD plus. So the NADH is here, this is reduced form, this is the oxidized form. So the energy present here are actually releasing in the form of the ATP. That will be determined from the P/O ratio or the P/2 electron ratio. O means this is the oxygen atom. One oxygen atom is taking 2 electrons. So we know that NADH is involved in the pumping 10 proton molecules across the inner mitochondrial membrane, and the succinate because it is taking the electron a step later. So there will be the 6 proton molecules that pumped into the intermembrane space. So 4 protons for the synthesis of an ATP molecule, and 1 is used for transporting the inorganic phosphate ATP across the mitochondrial membrane. So the proton base P/O ratio for the NADH is 2.5, and for the succinate is 1.5. How? 10/4 equal to 2.5, 6/4 equal to 1.5. Though in means most of the time you will see the P/O ratio of the NADH is 3 and P/O ratio of the succinate is 2. Okay. So this is how the proton pump is helping in the other function. So here how the ADP is moving into the matrix of the mitochondria. So the ADP is moving into it and ATP is exporting. This is an antipode system. Okay. So inorganic phosphate this is the proton that is being used in syn-coating. This is the phosphate translocase, and this is the adenine nucleotide translocase. This is an antipode. So it takes ATP generated in the matrix out of the inner mitochondrial membrane, and it means return one ATP molecule into the matrix. And the phosphate translocase take a inorganic phosphate from the inner mitochondrial membrane or from the outside into the matrix by a seam port mechanism using one proton molecule. So there is a proton here, and when this is the ATP synthesis or the F0F1 complex, when this is moving, the ATP transported here, inorganic phosphate transported here, they are combining, as I told you, alpha, beta conformation, and the ATP is released, that is moving out, and during this process, one ATP is going in. So we know that there is a synthesis of the NADH in the cytoplasm, and inner mitochondrial membrane is impermeable to the NADH. So how the NADH is reaching the matrix of the mitochondria, where it is donating its electron to the electron transport system. So here you can see that oxaloacetate, this is achieving through the process of the malate-aspartate shuttle. So NADH equivalent present in the cytoplasm. By this process is creating equivalent NADH in the matrix of the mitochondria without taking NADH into the matrix of the mitochondria. So this is the oxaloacetate. During the formation of the malate, it uses one NADH. So one NADH is used in the cytoplasm. This malate, it is moving by antipode system. Alpha ketoglutarate here is coming out, and this is taking one malate inside. So it is an antipode system of the alpha ketoglutarate and the malate. And in the matrix, this malate is releasing this NADH and getting converted to oxaloacetate in the matrix of the mitochondria. And this oxaloacetate, it is using one glutamate and converting the glutamate into alpha ketoglutarate that will be exported out, and oxaloacetate is converting to aspartate, and that is going out by another antipode mechanism of taking one glutamate in. So by this process, malate and alpha ketoglutarate, they are exchanging themselves, and they are counterbalanced by the glutamate coming in and aspartate going out. In the process, there is the equivalent amount of NADH that is used in the cytoplasm are generated in the matrix. So like this, the NADH that is generated in the cytoplasm shuttle into the matrix by the malate-aspartate shuttle mechanism. So there is another one. In case of the skeletal muscle and the brain, the NADH that is generated during the glycolysis, it is not going into the matrix. So the NADH is using, means it is getting oxidized and transferring its energy into the dihydroxyacetone phosphate to form a glycerol 3-phosphate. And this glycerol 3-phosphate, this is used by the mitochondrial glycerol 3-phosphate dehydrogenase in converting FAD to FADH2, that will donate its electron to the quinone to form the quinol. So here we can see there is a 4 less number of the protons are transported because this is bypassing the complex one, but this is enough for the skeletal muscles and the brain. So in case of the skeletal muscles and the brain, the NADH generated in the cytoplasm are transferring the energy into the quinone directly, so that it is going to the complex 3. So the first one is the glutamate aspartate shuttle. This is the glycerol 3-phosphate shuttle that is found in the skeletal muscle and the brain. Then in case of the plant, the NADH dehydrogenase, that is the complex one, that is externally oriented. So in this case, you can see that NADPH is there, that is converted, that is oxidized into the NADP, and the electron is released, means it is using in reducing the quinone to for the quinol. Okay, and that is moving into the complex 3 and the complex 4. And during this process, there is a pumping of the proton from the matrix into the intermembrane space. So we have the 2 shuttle mechanism, and in case of the plant, it is a special type of NADH dehydrogenase, the complex one. That is the externally oriented one in case of the plant. So now, based on this electron system, we'll calculate the number of the ATP generated from the one molecule of the glucose. So during the process of the glycolysis, There is the 2-NADH. So this is the reducing substance, and the ETS, this will be used in generating 3 or 5 ATP. And there is the direct 2 ATP generation of glycolysis. Then the pyruvate oxidation, there is the 2-NADH that will be used in the ATS to generate the 5 ATP. Then the acetyl coenzyme A in acetic acid cycle, that will generate the 6 NADH. So there will be the 3 for NADH. So for the 6 there will be 15, and there is the 2 FADH2. So for FADH2 there is 1.5, so there will be 3. So in glucose oxidation, one glucose molecule leading to the 30 or the 32 ATP synthesis, the final product. Next is the photosynthesis, because the electron, the way it transfers and releases its energy that is used in creating a proton motive force across the inner mitochondrial membrane, or in case of the chloroplast, that is similar. So we are taking a comparative study of the in the electron transport system of the chloroplast. So we know this is the sun, solar energy, the source of the energy is the light. The photosynthetic cell, they will fix the carbon to form the carbohydrate, and they will generate the oxygen. And entire heterotrophic environment, the ecology is thriving from the energy generated, energy captured by the photosynthetic cell in the form of the chemical energy, that is the source is the sun. So here the source of the electron is the water, not the NADH in case of the photosynthesis. In case of the mitochondria, it is the NADH. Here the source of the electron is the water. The end product of the mitochondria electron transport is the water molecule. Here the end product is the O2 molecule. Okay, so this is the two difference. And so this is a light reaction. In the light reaction, the NADP, this is the oxidized form, it will capture in the form of the NADPH. Okay, and there will be a synthesis of an ATP also. And finally, this chemical energy used in the dark reaction to fix the carbon in the form of the carbohydrate. So this is a plant cell. So in this we can see the chloroplast, and this is the chloroplast is also a bilayer, means the two phospholipid bilayer, there is a space between them, but this is not the inner membrane of the chloroplast that is having the electron transport system. The electron transport system is present in the membrane of the thylakoids. So these thylakoids are the single membrane structures, okay, and the lumen of these thylakoids are like the intermembrane space of the mitochondria. So the similarity of the thylakoid and the intermembrane space of the mitochondria. The inner membrane is equivalent to the membrane of the thylakoids of the chloroplast. So here the light is there, water is the source of the electron, the end product is the O2 molecule, there is the light reaction, and in the dark reaction, this chemical energy is used in fixing the CO2, so that the carbohydrates are produced. So this is the electron transport system, this is the thylakoid membrane, this is the stroma, means it is like the cytosol in case of the animal cells, and the thylakoid space is the intermembrane space. Okay. So this is the matrix. Stroma is like the matrix, not the cytosol, it is a matrix. So the light energy, this is the photosystem 2. Then this is plastoquinone. Then this is the cytochrome B6 complex that is reduced, and during this energy is released in creating this proton motive force, and this is used in generating the ATP. This is a very simple explanation. So what is this one? Photosystem 2 system. So this is thylakoid space, the photosystem 2 reaction center. Light energy is used in, and the electron is from the water molecules. Okay, in this process, one oxygen molecule is released and the 4H plus. Okay, and in this process, the quinone is reduced in the quinol, and this is the manganese cluster water splitting enzyme. So this is the enzyme that splits the water into the oxygen molecules and the H plus, and this electron travels through plastoquinone. And this is the energy that is released from this electron. They will reduce the ferredoxine. Ferredoxine will reduce. Ferredoxine will reduce the ferredoxine NADPH. reductage. Okay, so finally the NADPH is formed. So this is a slightly elaborate explanation of the ETS. So photosystem 2 we have already discussed. Now we are going to discuss the photosystem 1. Okay, so here this is the reduction potential. So in the photosystem 2 it is almost minus 600 millivolt, and this is a series of oxidation reduction process with the quinol, finally plus 2 cyanine. In this process there is the proton, the energy that is stored here, it is released when it is moving from the low reduction potential to the high reduction potential. The energy is used in pumping the proton from the stroma of the chloroplast into the thylakoid space. Okay, so this is the cytochrome B6 F complex. Then because of the presence of the light again, the reduction potential of this plastocyanin is further reducing to the less than minus 1200 millivolt, and in this process through a series of oxidation reduction process it is releasing its energy, and the energy is trapped by reducing the NADP plus into the NADPH. So in this entire process, we are getting NADPH, and where the light energy is stored in the form of the chemical energy. At the same time, there is the proton motive force is generated across the thylakoid membrane, which will generate the ATP as discussed in the mitochondrial electron transport system. The proton will move to the F0 F1 complex, and through that alpha beta means ATP binding conformation, ADP plus inorganic phosphate, loosely binding conformation, and the beta empty conformation passage of the proton. There will be the release of the ATP into the stroma of the chloroplast. Thank you.